So what does the inside of the liver look like? Well, it looks very different from the rest of the digestive system because remember, it's not part of the gastrointestinal tract. It's an accessory organ. It's a filter. So the structure, the histology is going to be very different. The connective tissue separates the liver tissue into these little hexagonal shapes that we call lobules. So the lobes are divided into lobules. And that's an organization we're going to see again in other organs. The cells in the liver are called hepatocytes, which literally means liver cells. Blood enters at the corners of each of the lobules through a branch of the hepatic portal vein, a branch of the hepatic artery, and then the oxygenated blood from the artery, the deoxygenated blood from the vein pour through sinusoids that branch off of both of those. And both of those kinds of blood mix. So you get a mix of oxygenated and deoxygenated blood flowing past the hepatocytes. So these cells get oxygenated and at the same time can do their job of filtering toxins. The blood then drains across the lobule to the center and exits through this central vein. And each of the lobules has a central vein at its, center. Now there's one more tube here. Come back to that. So here is a close-up on a portion of the lobe. These three together are called the portal triad. So it's the branch of the hepatic portal vein, the branch of the artery, and then a bile ductule. Because remember, one of the functions of the liver is to make bile. So it's the hepatocytes here that actually make the bile, and they secrete it into these tiny little tubes that run between the cells called bile canaliculi. And you remember canaliculi just means little canals. Those canaliculi carry the bile out to the bile ductule. Okay, so the bile flows this way, the blood flows that way. Now you remember the hepatic portal circulation. We had the blood from the small and large intestine, the inferior mesenteric vein, the superior mesenteric vein, the splenic vein, all merged together to create the hepatic portal vein. The hepatic artery proper branches off of the common hepatic artery, which branches from the celiac trunk. Okay? Both of these are going to branch as they move through the liver, taking blood to the lobules. That blood goes through across the lobule, exits through the central vein, and then the central veins join together to create hepatic veins, and the right and left hepatic veins then take the blood to the inferior vena cava. So that should all be review except the part about the lobules. Okay, so the portal triad is these three here. And they are going to be located at each of the 6 corners of the lobule. The capillaries within the lobule are sinusoids. And remember, sinusoids have big holes in the wall because these hepatocytes need to not just filter out toxins, they need to evaluate the health of red blood cells. So they're going to allow red blood cells to leak out so that they can be evaluated by these hepatocytes. The other thing we see here are these reticuloendothelial cells, which are cells along the walls of the sinusoids that are immune cells. They are phagocytes. They're going to engulf and break down any pathogen that might make it this far. The hepatocytes absorb nutrients, produce bile. The bile then is carried out the bile canaliculi to the bile ductule. The bile ductules then fuse into a bile duct which carries the bile to the gall bladder. What that looks like on our models, here's a lobule. You can see it's hexagonal. There's a portal triad at the corner. Again, this is just a portion of the lobule. Here's a portal triad, and then each of the lobules has a central vein at the center. These are the sinusoids, which you can see more clearly here with the reticuloendothelial cells in the walls. Those are those immune cells. The different things in the portal triad, the hepatic artery, hepatic portal vein branches, and then the bile ductule. The cells are hepatocytes. And on our next section, we're going to talk about the pathologies of the liver.